Hello, good afternoon. This is Raj Agarwal. If you don't know me, I am with the Cancer Center, co-leader of the Prevention Control Program, and it's my immense honor to introduce our speaker today, Sean Davis. So, Sean joined us in 2021, which is this year only, a few months back actually, as a professor and chair of the Cancer Informatics. He's also our deputy director for the Center for Health Artificial Intelligence, as chief scientist from NCI till 2020, and before coming here. Sean did his MD PhD from University of Pittsburgh, and I don't hold him for being esteemed friend, but part of the package when he joined here is that he will convert to the Broncos friend. So he's my Broncos friend, I hope. Sean has enormous amount of honors, awards, several funding, invited speaker all over the globe, 100 publications. So I can keep going on and on and on, but I don't want to take his assigned speaking time because we are super, lucky that Sean joined our university. So Sean, it's all you now, please. And his title is right over here. Thank you.
All right. Well, thanks for the invitation. Thanks, Raj, for the great introduction. Looking forward to chatting. And for those of you who are on Zoom, we have an enormous audience here of at least 6 people. And I'm waving to all of them one at a time now. We'll hopefully have some interaction. And for those of you at home, following along at home, there's some hands-on coding. So for those of you who are bioinformatics practitioners, we can go through some of this, depending on how much time we have. I'll do some live coding in front of you here. The topic that I'm going to be talking about is Bioconductor. It's a labor of love. I've been involved with the project for about 15 years, and I'm talking about the project without telling you about it. I'll come to that in a bit. But the focus of this talk is going to be on increasing the value of public data through software and data engineering. So Eduardo asked me to give a research talk, and some people might think of software engineering and data engineering as not research. And we can have that argument, but at the end of the day, I feel like the kind of work that the Bioconductor project does is not just about the research that we do, but also about the research that we facilitate. For those following along, I should mention the slides. The link to the slides is in the bottom right, or you can use the little image to get to the URL for the slides. And then the code examples are going to be linked here. So you can click on that link, and that will take you to the actual code that you can use to cut and paste. So the problem statement for Bioconductor. Bioconductor started back in 2001, and it was developed. The project was developed by some folks at Harvard to establish reusable, interoperable, fit-for-purpose software tools that are required to input, manipulate, evaluate, integrate, and comprehend microarray data. So microarrays, when I joined my group in 2002, microarrays were brand new. And the idea of being able to get more than like 3 genes worth of gene expression from a machine was remarkable. So this was, microarrays generated the largest amount of data that most biologists had ever seen. And so there was a need for new tools to analyze those data, to understand them, to work with them, and then finally to comprehend them, right? And that's what biological or biomedical research is all about. The problem statement in 2021 is roughly the same, except that we're not talking about microarrays anymore. We're talking about all of this. So, and this list of assays is not chosen at random. These are assays that particular software packages in Bioconductor say specifically that they deal with. So when I put this list up here, it's not made up. There is at least one software package to deal with each of these, and in many cases, there are dozens. So what about software, and why is software so cool? Well, and this is my statement about software. Software encodes knowledge. So if I can write something down on paper, this is the way I did it, that's great. I've transferred my knowledge to paper, but somebody still has to interpret that, those words. So a standard operating procedure, unless it's written really, really carefully, doesn't actually capture all that's needed to reproduce some work. Software does. Software is the actual recipe. It includes all the ingredients as well, and it's highly reusable and reproducible, especially if it's open source. That means that we can share it openly. So software, if it's written well, encodes the knowledge that the software writer included or knew, but often it encodes it in a way that simplifies the use of that knowledge. So we'll get to that in a bit. What is Bioconductor? Well, Bioconductor is a large, and when I say large, I should capitalize it. We'll get to the numbers in a second. It's a large NIH-funded open source software community dedicated to the analysis and comprehension of high throughput biological data. And I've highlighted a couple things. It's NIH funded. The NIH funding, though, for the core Bioconductor project is really just a couple of grants. So it's about $2.5 million a year. And we'll get to the numbers. That's a pretty good return on investment in my opinion. It's a community. So Bioconductor is not about a set of PIs who do this work. This is about people contributing their time, sometimes for free, sometimes nights and weekends, but sometimes, as part of a formal research project, contributing that knowledge, that software to Bioconductor. And analysis and comprehension. This is all about research. It's not just about software that does stuff. This is how people actually do research with data. Bioconductor, by the numbers, started in 2001, 2002. Bioconductor is built on top of a language called R. R is a general purpose statistical software environment. There's an estimated 2 to 3 million users of R in the world. And what that means in practice is that it's not too hard to find people who know how to use R. It may be hard to find biologists who know how to use R, but R is pretty ubiquitous. R has, these are old numbers, 15,000 or so contributed packages. And Bioconductor is a piece of that is biologically related software, has a couple more thousand. Bioconductor gets about 55,000 downloads per month, and that's not exactly equivalent to users, but it's roughly equivalent to people who are trying or using a software. So 55,000 users per month. And those are unique users, so we're getting new people all the time. There are more than 22,000 PubMed Central citations, and that's just to the core Bioconductor sort of marker papers. If you include all the software packages that are written up in the literature, it's probably in the several 100,000 range. Bioconductor is released twice a year, and released means that we make the software, a new version of the software available twice a year. And as of the last release, there are 2,042 individual biomedical and omics related data science software packages. Each of those has been contributed by one or more people, so a team in some cases, and those people are maintaining these software packages for now and the future. Some of them have been maintained for 20 years successfully. We receive submissions of about 3 to 6 new software packages per week. So that's how Bioconductor grows. And there are hundreds of active developers and thousands of developers who are attached to these packages. I'm now going to read all of these software package names. That's when the group here is supposed to laugh, but no one's laughing. Okay, moving on. Bioconductor has many capabilities, but I'll just say that Bioconductor is used by people every day. It's used by its developers, and it's used by end users who are not developers. And it supports all the phases of biomedical data science workflow, everything from specialized import and output, or import and export, to data management, to visualization, to reproducibility, including people in the loop. So this is not the kind of thing that you run as a pipeline or a workflow. This is an environment for engaging with your data. And finally, Bioconductor has built in lots of capabilities for not just being reproducible itself, but developing reproducible research and publishing reproducible research. And we'll see an example of that in a bit, many examples of that in a bit. This is Bioconductor contributions over time. This stops in 2018. I haven't updated the slide. The blue fit, or the blue curve, is an exponential fit. And by exponential fit, I mean there's an exponential equation underlying it. You guys can make your decision about whether or not Bioconductor contributions have been increasing exponentially, but I think it'd be hard to argue that that's not a close fit. A core value for Bioconductor is reproducible research. That means a lot of different things to a lot of different people, but I'm not going to go through some of the horror stories in biomedical research and with clinical context where people have made mistakes based on either not using reproducible research techniques for computational research, or something as simple as having a cut and paste error in Excel that actually made its way into a clinical trial. So part of what Bioconductor aims to do is to enhance reproducible research. And what I'm showing on the right-hand side of the slide here is one sort of core capability in Bioconductor, which is a so-called data container. So data coming out of biological experiments is pretty complicated, right? You might think of it as a set of numbers associated with a sample, but as soon as you start adding samples, it gets a little more complicated. Each of those samples typically has tens or hundreds of things that we know about it. So there's another axis or another dimension. And then finally, for the features that we measure, whether they're genes or proteins or chromosome locations, each of those features also has metadata, things that we know about it. And if you think about how you might typically model those data, you'd put them into Excel and you'd have a bunch of columns and column headers. And sometimes you might even have multiple Excel spreadsheets that have to be related to each other. Well, Bioconductor recognized this is a challenge, and so built specialized containers for biological data. And what you're seeing here is in the middle, the assays. Those are the measurements. Those are the Excel spreadsheets. And then the column names in the Excel spreadsheet, we extend to allow essentially multiple column names or multiple pieces of information about each column. And that's the call data in the upper right-hand corner. And then the row data is information about the genes. So we have a gene name, for example, but we also might know that a gene has a specific function or has a specific chromosome location or has a set of different names. All of those things are the row data on the left-hand side. And if we put all these things together, we have something called a summarized experiment. And this is kind of cool because it helps us to track our data. It helps us to share our data with all the complexity that comes with it. But it also allows us to do stuff like this, where we take a summarized experiment sitting in the middle here. That's our description of our data. If we can bring in data from any resource and get it into a summarized experiment, then we're essentially starting in Bioconductor with the same format, the same information content in one object. And from there, we can do things like on the left, pathway analysis, bottom left, plotting along the genome, middle on the bottom, a t-SNE. On the bottom right, differential expression analysis, and on the top right, principal components analysis. With the same data structure, this summarized experiment that encapsulates our biological experiment, we can jump immediately, based on other Bioconductor packages, interoperability to all of these different functionalities. And because we start with these common data structures, we allow a lot of reuse as well, code reuse as well, code and data reuse as well. Bioconductor is pretty connected to the rest of the world. Putting Bioconductor in the middle here, it doesn't have to be in the middle, but all of the boxes around the outside here are other data resources or analysis resources. Some of you might recognize some of them. There are probably many others that you don't recognize that are, or that you know that are not on here that Bioconductor connects to. But when I say connects to, I mean literally there's a bridge between this thing, this data structure, and these data resources. So we pull the data out of these data resources and load it into that data structure. And now we're back to being able to do this. So public data, and this is sort of the jumping off point. We're talking about bridges between public data resources and Bioconductor capabilities. And I'm going to focus on the bridges themselves and not the downstream analyses after you get the data into Bioconductor. There's tons of applications for all kinds of things, and they're all well documented. I didn't mention that, but all Bioconductor packages, all 2,042 of them, are required to have a so-called long-form documentation. So basically, a tutorial on how to use their capabilities. The first one I'm going to talk about is the gene expression omnibus. This is the reason I talk about this is that this is how I got hooked in Bioconductor. I was using Bioconductor for about 5 years before being asked really to contribute, and I got a phone call from Robert Gentleman, who was one of the founders of Bioconductor, and also the person who wrote R, called me out of the blue. Sean, I see you're answering stuff on our email list. That's how I really got involved with Bioconductor, is answering emails. You're answering stuff on our email list, but there's these folks at NIH, these folks at NCBI Geo, and you're at NIH too, right? Can you go and talk to them? Because they think that we should be connected to them. So I went and talked to them, and we, came up with a, big project plan, et cetera. And I went home and started playing around a little bit and realized that actually within a couple of weeks, I could probably have this done. So GeoQuery came around, and what does GeoQuery do? Well, in the bottom right-hand corner, maybe a little hard to see, there are 4,684,836 publicly available gene expression data sets for free, right? If we think about the cost, let's say it's $100 for a sample. I have no idea if that's the right number. We're talking about $400 million worth of data publicly available and free to you. And so we're going to try to make that really easy to access and then analyze using Bioconductor. I'm going to use a really, this is a cancer talk, so we'll use a driving example. This is the paper that we're going to look at. Now, my use case is I'm going to the literature, I'm searching for a paper, I find a paper that looks interesting to me, I look it up, I find it on PubMed, I scroll down a little bit on the right-hand side, I'm not going to show you how to do that, but on the right-hand side there's a link to related information, and there's a little link there called geodatasets. So if a paper that you find has a link to geodatasets, then here's where we go next. We jump over to Geo, another database at NCBI. This is the database that contains essentially functional genomics data. It's ChIP-seq, RNA-seq, and microarray gene expression, methylation, things like that. A lot of samples, as mentioned. So here's the record for that paper, for the data from that paper. And you might say, well, I can just go and download it, right? Well, here's what would happen if you download it. You get this. So I know it's a little hard to read. Even if you got closer, it's still hard to read. So when I say software encodes knowledge, all I did was to figure out what this format was and write a function that can allow us in about 10 lines of code to generate this plot from those data. Here's what the code looks like. And I'm just in the, let's see. I'm gonna go ahead and do one example by hand, or one example, code example, just so that people who haven't thought about R and Bioconductor can see what it actually looks like to do. So I'm gonna jump over to something called RStudio. And what's going on here on your screen right now, let me make this bigger. What you're seeing on your screen right now, or on our screens right now, is how we use R. There's no GUI, there's no click to use GeoQuery. This is all text. It's all text, and that's not a bad thing, it's a good thing. Because what I'm going to show you, if you install R on your computer and you do this, you type the things that I'm typing here, you will get exactly the same result. That's what software is all about. And reproducible research is about being able to write down the recipe exactly so that you can share it and reproduce it. So I'm going to use some shortcuts here, but in the upper left-hand corner is the code that I'm actually going to be executing. And in the bottom is where the code is going to show up. So I've loaded the GeoQuery library into R. And I need to connect to the internet. That's always a good start. Apologies. This is so you guys know it's actually live. All right. So what I'm doing now, what I asked GeoQuery to do with the getGeo function is to go and grab the data that we saw associated with that paper. It's parsing those data into R. And then I'm going to convert this. This is a detail that you don't really need to follow along with. I'm going to convert this, though, into that summarized experiment. Remember those colored blocks on the screen, the data container? The summarized experiment is the data container. And I've just converted the data that I got associated with that paper into that, that summarized experiment type of container. And what I just did was to, I knew a little bit about this experiment, but what I just did was to ask our GeoQuery Bioconductor to summarize the metadata about the samples. And in the columns, we have whether the sample is representing normal or tumor, and down the column or down the rows, the type of tumor. So, for example, for the first row, we have 65 no's. That's 65 not normal. So these are 65 tumors, and 10 yeses. So 10 normal samples for our breast cancer. And the same thing, 57, 12, et cetera. So if you look through, we can actually, if I type SE here, you can see that I have measurements. There's a little thing there called DIM. I have 54,715 genes that I got, and 280 samples. So that's the data set that we're working with. And it has all the information about the genes, all the information about the samples, and we looked at how we could get a little bit of that information about samples and make a table out of it to see what's there. So now what I'm going to do is, this is now moving away from GeoQuery. This is now using Bioconductor and R capabilities. I'm going to make a principal components or multidimensional scaling plot. And I'm not going to go through what that is, but I'm going to choose genes that are the most interesting, that is the most variable, the ones that change the most across the experiments. And the next few lines here are to just generate the principal components analysis. You can see I'm doing this in real time. So you could actually play with this, with these analyses as you see fit. And here's what you get. And let's see if I can do this. So it's a little bit hard to read here, but the different colors represent the different types of tumors: breast cancer, colorectal cancer, non-squamous cell lung cancer, and prostate cancer. And then this is actually really hard to see, but it's there. Tumors are circles, and normal tissue associated with those tumors, adjacent normal associated with those tumors, is a triangle. And you might say, oh, look, the tumors separate from each other. Cool, except look, the normals do too, right? So what's the hypothesis that we just generated here? Well, that these tumors are different from each other, not because they're different kinds of tumors, but because they come from different anatomy. They come from different tissue sources. So With 10 lines of code, and I don't know, I looked at my watch, it took me 5 minutes to do that analysis. And if you might say, well, Sean, I don't know how to do that. Well, I gave you the recipe with the code. So you can follow along with the code and reproduce this exact result without knowing very much about Bioconductor. All right. So again, I just wanted to show you sort of a feel for how Bioconductor works in practice. It is text-based, and it is, again, meant to be reproducible, reusable. I saw a pencil in the air, so are there questions? Okay. Any questions? All right, cool. Let me go back to slides here. I'm going to move on to specifically different public data resources now. NCBI Geo is what we spend our time talking about. It's about 4.5 million samples, and general those are general functional genomics data from all different species, from all different diseases and normal biology. What we're going to be talking about now is curated cancer data. This is human data, and it's coming from sort of these enterprise cancer resources, the TCGA data, and also CBio portal. So there's a great paper from for those who are interested, sort of the details of this, called Multi-Omic Integration of Public Oncology Databases in Bioconductor. It's chock full of examples and is a review article really on all the cancer-related data resources and tools in Bioconductor. This is a slide just showing that there's a spectrum of capabilities in Bioconductor that go from high integration capability and high ease of use to very complete, but maybe a little bit more difficult to use. And for those of us who spend our days thinking about how to use cancer genomics data, sometimes we actually need to get down to the completeness and maybe not so easy to use in order to do some of the analyses that we'd like to do. I'll go through examples of sort of each level here. So you might say, well, why use Bioconductor to access TCGA data? Well, think of it in the same way that I showed you the picture of what the geo data actually look like in the database. Multiply that complexity quite a bit, and you have cancer genomics data. So in the bottom right-hand corner, we see all the different platforms or assay types that are represented. And this is just TCGA. 50 different platform types in TCGA, 33 different diseases, and 19 different data types. So you could figure all these out yourself, or you could use what Marcel has put together, a couple of tools, curated TCGA data and CBio portal data. We'll go through those here. So what does this look like in practice? Well, on the slide in front of you, there's literally 3 lines of code to type. The stuff that's in blue is the output of these commands. So we can ask, for example, with 1 line to get all the curated TCGA data, all the data from TCGA associated with adrenocortical carcinoma, ACC. 1 line gets us all that. We don't have to go look at a website. We don't have to figure out what downloads are available. It's all done for us. And the curated part, I didn't mention, the curated part is that all of these have been hand curated. So there are actually mistakes in TCGA, people that have been assigned the wrong sex or duplicate samples. Those have largely been corrected, but the curation process that Levi Waldron's group and Marcel has put together corrects a lot of those. If we want to get the data, so on the left-hand side, we're getting a list of the data that are available. If we actually want to get the data, we use the code on the right-hand side, and we just essentially ask for it. We ask for the data for adrenocortical carcinoma from reverse protein phase arrays or RNA-seq. When we hit enter, curated TCGA data goes out, gets those data, the curated form of them, and pulls them back into these data containers that we've already looked at. you could go this workflow and jump directly back to that multi-dimensional scaling, that principal components workflow, because these data sets are encapsulated in the same kinds of data containers. So we replace the data from the PubMed entry with the data from curated TCGA data, and now we're off to the races again. TCGA, it's big and very thorough, but there are hundreds of other omics data resources or data sets associated with cancer. And some of these, actually more than 260, have been loaded into cBioportal. You can click, and probably a lot of you have used cBioportal. What cBioportal data does is to do exactly the same thing. It goes out to cBioportal and pulls those data into R. If cBioportal does what you need it to, makes the kinds of plots, does the kinds of analyses that you want, great. Don't bother looking at this slide. But if it doesn't, and you want to do something different with the data, you can, again, use the code on the left here to go through. And in this case, we're pulling exactly the same data, adrenocortical carcinoma, reverse protein phase arrays, and actually in this case, copy number analysis. And at the bottom here, there's a little trick for those of you who don't want to work in R and would rather work in Python. There's a little function that allows us to export these results as CSV files. NCI has built its own data portal, the Genomic Data Commons, and the Genomic Data Commons is great in the sense that it has a really nice interface. You can search, you can find data resources. But if you wanted to do this on dozens or hundreds of data sets, or if you wanted to do this in a very reproducible kind of way, you want to be able to take your analysis and share it with someone, you can't tell them where to click. You can't show them all the clicks that you did to get to the data, how to download it, how to analyze it. So we built a small package called the Genomic Data Commons package that is the bridge between the Genomic Data Commons and Bioconductor. We've been talking about gene expression data, those kinds of things. I thought I'd switch gears a little bit and talk about somatic variants. So our goal here is to explore the somatic variants in cutaneous melanoma in a reproducible and reusable way. Here's the code. This is all the code that you need to reproduce the analysis that I'm going to do. I'm going to show you the relationship between the code and what's on the website, just so that you can see it. We can do these simultaneously, or we can do these separately, but this approach gives us an entirely reproducible workflow. So we're gonna generate this plot. So the first thing we need to do is just find files. So we're going to use the files command. And on the website, the files command gets us to this page, and we're going to choose TCGA cutaneous melanoma. I'm going to select the same thing, TCGA-SKCM, using code. I'm going to pick somatic variants. Here's how I do it on the website. Here's how I do it in code. This gives me, in this case, just one file. I can get the ID for that file. And then on the website, I download it essentially. Here I use GDC data. That's going to get me that file. And now I'm going to jump to use one of those built-in Bioconductor packages that isn't built for working with GDC somatic variants. It's built for working with somatic variants. So how do we bridge between the somatic variants that we got from the Genomic Data Commons website through our code into this math tools package? Well, we got a file that was called a math file. We're going to load it. We're going to use read.math, and we're going to feed it the file name that we just downloaded. And again, you can see here, for those of you who are code, I'm using variables here. So if you wanted to change this to use breast cancer instead of cutaneous melanoma, you change at the top to use breast cancer. And with one more line, literally one more line, I get this plot. And with math tools, we can do a few more things. We can do actually many more things because math tools is built specifically for analyzing somatic mutation data. Saw this already. We can look at mutation numbers in clusters. We can look at specific genes. This is looks like BRAF. And if you didn't know, there's a specific BRAF mutation that's often associated with cutaneous melanoma. 198 samples out of, it's 49.89% of the samples. We have also, I've been working with Levi Waldron's group on UO1 for the last couple of years, looking at metagenomic data. And specifically, the curation process here is toward metagenomic data that are associated with cancer. We are now processing, we've processed about 22,000 or so samples. There are probably 400,000 samples available in metagenomics associated with human disease. And so I've built a high throughput pipeline for analyzing those data in a homogeneous way on cloud resources. For those of you who are interested in pharmacogenomics, and in particular in high throughput, high content screening of some fairly large NIH-funded projects, there's the PharmacoGX package. For those of you who do that kind of thing, on the the upper right-hand side, you can see the different data sets that are available: CCLE, GCSI, GDSC. These are probably names that folks who think about pharmacogenomics and cancer recognize. And some relatively recent work that I'm not going to go into because of time is a package for taking all the publicly available, in this case, up to this point, human data, and looking for shared signals across them, shared biological signals. So we have about 1.5 million human human samples that have been homogenously analyzed. And what we do is to look across all of those samples to find underlying biological signal. We use principal components for that and look for shared principal components. And that allows us to do all kinds of fun jumping between new data, any even rare disease data, back to publicly available data, and of course the literature associated with those data. There's a poster noted here. There's a preprint, and it's been submitted for publication. For those who want to dive a little bit deeper, one thing that Bioconductor has done in terms of building its community is we've hosted an annual conference once a year in the summer usually. That's actually expanded now to 3 different conferences, one in Europe, one in Asia. The one in Asia is actually taught in multiple languages: Mandarin, Chinese, Japanese, and English. And that conference has grown to well over 1,000 participants. And about 50% of those conferences are devoted to teaching hands-on workshops. And the hands-on workshop for these public data resources and a few others are available here. And for those of you who don't want to who don't want to install R and do that kind of work, there's a public data there's a platform that we use for doing these hands-on trainings. We've taught up to 600 people simultaneously on 5 continents in these workshops, and we've taught about 7,000 or so such workshops, including workshops specifically devoted to data science in Africa. And here's the platform for doing it. And one of the actually, I can just show you. Click out here for a sec. Public data resources in Bioconductor. If I click launch, then what I will get in about a minute, or a little less, is an RStudio workshop that it contains all of the code and installed software that you could use in your web browser to go through that public data tutorial. I'm not going to wait for that to finish, but We have, it's a big community now. We have some governance built in, a technical advisory board, a relatively new community advisory board. We have a scientific advisory board. We have developer forums. We have Slack. We have now formal working groups and many other capabilities for building, around building an equitable and inclusive community. So I'll stop there. Any questions? Oh, wow. I'm overwhelmed with the applause.
Thanks, Sean, for the enlightening presentation. From the audience here, any questions? How the virtual system works?
They can unmute themselves and ask questions.
Okay.
Yeah, you can tell them they can unmute themselves and ask questions.
Oh, sorry. I guess we're now at the unmute and ask questions phase.
If they have questions.
If you have questions. All right. All right, crystal clear. Awesome. So feel free to reach out to me if you have questions or, oh.
So, I mean, where do you see this whole program forward going? Depends on research. So, the original program, we have a lot of things that, like that. If we want to do some of this with your expertise, where do you see us moving in terms of some collaborations?
Yeah, so Bioconductor I guess I wasn't entirely clear about this. So the question is, how do we use it? How do we get involved with it? So Bioconductor is free, it's open source, and for someone who wants to try it and use it, the website bioconductor.org has very explicit installation instructions. In terms of collaborations, there are. I would guess that anyone who's operating with genomic data is already using Bioconductor. That would, if you ask someone who is working with genomic data, who's a bioinformatician or a genomic data scientist, they probably will be using Bioconductor already. And that is not just here in Colorado. That's anywhere in the world. Yeah. Yeah.
I would comment on that.
Yeah.
Year by year, our students are getting more literate with being able to do basic manipulations in R, Python, whatever. And the generation is coming up. This kind of thing provides a real service because it gives you kind of a one-size-fits-all access to lots of different genetics data and all the different software packages that you might want to use to manipulate it. And the beauty of what you're talking about is that it's whatever you want to access, it's all with the same structure. You don't have to reinvent the wheel every time you go in. And so with a little bit of basic facility in working in R and a resource like this, you can do almost anything.
Yeah, that's so. Thank you. I think that told me about about clarity. So the comment, it wasn't a question, it was a comment about how a Bioconductor facilitates science by sort of crystallizing a common language and data structures and connects different data resources together so that you can build an integrated analysis. And the other piece of that comment was that students and, well, just in general, the workforce has gotten a lot more fluent with using computational approaches. It is part of not even formal training, but it becomes something a lot of folks who are coming out of high throughput biology groups have been exposed to. I'll jump off on that comment and just say that there's people ask me all the time, so how do I get started? Well, I have good news and bad news. The good news is that getting started is not that hard. But it also is also a little bit different than getting started with a lot of other things that we're used to learning. There's not a specific course called Learn Bioinformatics. There are those courses, but they won't do what they say. What you actually want to do is to try to think of the question that you want to answer. And then start delving into the resources that look like they're associated with that question. So cancer genomics and bioinformatics is not something you can ask a question about, but if you have a data set, you can begin with that data set. What are the questions that I want to ask, and how would I get started with asking those questions? We're adult learners, right? Most of us on this campus are adult learners. We don't have undergraduates who are taking a lot of courses, who are being fed information. We're adult learners. We want to know how to do things. We want to know why we're doing them. And we just want as quick a process to get there as possible. So adult learners need to sort of self-motivate to a certain extent. And one of the things that I think about when I teach bioinformatics is I'm not worried about teaching you skills. I want to teach you confidence. So the goal of teaching adult learners is to give them enough skills so that they know where to go for the next question. But this may sound foreign, but people who are coming into these kinds of fields are really uncomfortable asking questions. So one of the things that I do in a course that lasts longer than a day is I require students, or I ask of students, to go to an online forum and ask a question in public that addresses some particular concern of theirs. Once you learn to do that, once you get comfortable with asking a question, then all bets are off. You can literally then do whatever you want. So that's a long-winded answer to say there isn't necessarily a course that's going to teach all of this, but there's a lot of documentation available, and understanding what your use case is, what you want to get done is where to start, rather than I want to learn the tool. No, I want to answer a question. So, other questions, comments, follow-up?
Do you have anything on the virtual? Can you see this shop? Virtual?
I don't have access to it, so.
I can't see it, but no, there's no.
Okay, cool.
I put a message to the Nicola.
So he stays here for training now, or we can do it?
We're done if he's done, nobody signed up.
Cool.
All right, well, Sean, thank you very much for your time. And, this is kind of difficult process of doing invited seminars like this, which you did, and we appreciate it, because virtual is